Okay, I was making 7B2, and of course I cut it off at the wrong time. So these are the two shock waves that I wanted to show you. The first one's going to talk about translation, and the second one is going to actually give it to you in words, but I'll let this guy talk. I will stop at one place. I will give you that.
The genetic information stored in DNA is transcribed into messenger RNA and then translated into protein.
Okay, so this is where I'm going to stop it. It's important to remember that it went from going from DNA to mRNA. It's not a big switch. They're both nucleic acids. In fact, this is called deoxyribonucleic acid, and this is ribonucleic acid. Remember, this just got an oxygen away from a hydroxyl that kind of hits off the end, and this one just has the regular hydroxyl. So this DNA to RNA is just called transcription. It's just basically copying it from one form to another of the same form using the same language. And then translation is actually changing it from nucleotides to amino acids and proteins. And remember this nice little ribbon shape. And this is what the protein is trying to get. So let me go back.
Translation begins when messenger RNA binds to the ribosome. The initial transfer RNA occupies the P site on the ribosome. Subsequent transfer RNAs with bound amino acids first enter the ribosome at the A site. The complementary matching of 3 nucleotides on the transfer RNA, called the anticodon, and 3 nucleotides on the messenger RNA, called the codon, ensures the correct sequence of amino acids. The messenger RNA passes along the ribosome in short spurts of 3 nucleotides at a time. As this occurs, the initial transfer RNA is moved to the E site, and its amino acid is transferred to the second amino acid at the P site. At the same time, a new codon is presented at the A site. The initiating transfer RNA, which now no longer carries an amino acid, leaves the E site, and the next transfer RNA with a complementary anticodon enters the A site. Each time a new codon sequence moves into the A site, a new transfer RNA brings in an amino acid. The old transfer RNA, paired with the previous codon, is passed to the P site and then to the E site as the amino acid it carried is transferred to the growing amino acid chain. As the ribosome proceeds down the messenger RNA, a stop codon is finally encountered. At this point, the ribosomal complex falls apart and the protein is released into the cell.
And the ribosome is free to go over and find another messenger RNA, the large part and the small part. And the messenger RNA that was used to make this protein can actually go find another ribosome to make more proteins, all determined by what the cell needs or requires. Okay, both of these are shockwaves, so they won't work on Apple, but I'll post the links to both of these underneath this video when I post it. Okay, so now we actually have the same thing, except now it's going to be in words. We have the nucleus, we have chromosomes inside, we have these ribosomes. Remember, ribosomes can be free-floating in the cytoplasm like these, or they could actually be attached to endoplasmic reticulum, or ER, creating the rough type. Protein synthesis is making proteins. It uses information from the DNA, from the chromosomes and the genes. Proteins are very important because they actually make you, they make the cell, and they're actually put together with structures, enzymes, remember, which are proteins that do special things. Proteins are long chains of molecules made of amino acids. Different proteins are used, and I said I wasn't going to read it. So basically what we do is we take the genes off these chromosomes and we actually split it using an enzyme, DNA helicase, and it actually splits the 5'3" from the 3'5", and the 3'5" actually is the substrate that we're going to use to make the messenger RNA because we need to come out with a 5'3" RNA. Zoom in and take a look at it. There's the DNA strand. Helicase comes and unzips it. This is the unused side. This will be the gene side. So this is the 3'5" strand. And remember, we can't use, and I don't know which one's first, but they'll tell you. We can't use T's, thymine, reading with the A's. I'm going to assume it's this one is the A, but we'll see. Looks like that one is the A because this is the U, and they're saying that because it's purple. RNA, one strand of DNA, the other strand of DNA makes this strand makes the messenger RNA. Then the DNA comes back, and remember the RNA polymerase puts these nucleotides together, puts them together and if you take a look, puts them in threes, which will be called the codons. This messenger RNA leaves the nucleus, goes finds a ribosome, and then the ribosome will take that genetic information from the messenger RNA and help make a protein. Got codons. Messenger RNA hits the ribosome, brings in a tRNA, a transfer RNA with the anticodon, with the amino acid, starts off with methionine. So this is the AUG, which brings in this particular one, which brings in the MET amino acid. Two amino acids come in, get peptide bonded, make more than that. Now we have polypeptides, and this is the way we make proteins. And remember, they've got to have that funky ribbony swirl type thing too. And I think that is going to be, they are very specialized, and the messenger RNA can go find more ribosomes. The ribosome can read any messenger RNA that's given it, and it's all sped up with enzymes. Okay, that's going to be 7B2, let's call it B. Thank you.